Well, hello, hello, hello. Welcome to another afternoon at MSK Innovations. I'm your host, TJ Halverson. Today we have another great event lined up for you. Before we introduce our panel, I'd like to thank the sponsor of today's event, Orthopedics This Week. We have two really great companies coming to you today, Renovos and OrthoPreserve. On our panel, we have Aaron Smith, Chris Lyons, and Chance Leonard. Before we introduce the companies, I'll let Chris, Aaron, and Chance each take a moment to introduce themselves. We'll start with you, Aaron.
Great. Thanks for having me on, TJ. My name is Aaron Smith. I am now Managing Director of Vecteris MedTech Consulting, out to support the many companies in this space that I love so much. Been working in orthopedics now for 31 years, started as an engineer doing product design in the spine industry, moved through several different companies, big and small. Most recently, I was Chief Executive Officer of Artelon, company that was an aspiring market leader for chronic ankle instability implants. We sold Artelon to Stryker back in July of 2024. Really nice transaction for our investors, employees, all of our stakeholders. And now I'm hoping to bring some of that knowledge to other companies that can benefit from it. So
wonderful. Yeah, thanks for joining us. Excited to have you. Chance, quick introduction.
You bet. Good to see you, TJ and Drew, and fellow panelists and presenters. Really appreciate the opportunity to be here today. My name is Chance Leonard. I currently serve as Chief Executive Officer of Genesis Innovation Group. This is my 31st year in the medical device space, and probably like Aaron and Chris, we know that it goes really fast, but a lot of great companies to evaluate and provide input and feedback on. So really enjoy the opportunity to be here today.
Yeah, pleasure to have you as always. And Mr. Lyons, we'll let you give a quick introduction as well. Let's see if we have him.
I'm back.
You're back. There we go. Quick introduction.
Something happened.
Thank you.
Yeah, very quick. So Chris Lyons, been in the industry 36 years. Smith & Nephew for about 13 of that. Medtronic Spine for about 13 of that. M&A, heavy M&A at Medtronic Spine. And in the last 8 years, been running a successful Southern Metrics Consulting. I've had 3 very large exits that have totaled in value of about $650 million in the last 4. So excited to be here again.
Yeah, great to have you as always. Now, before I introduce our wonderful companies today, MSK Innovations, just a quick note, is not a broker-dealer nor an investment advisor. This is for informational purposes only. And with that, we'll jump into our first presentation with Agnieszka from Renovos. Agnieszka, take it away.
Thank you. Let me just try and share the screen again. And hopefully you can see that okay. Is that correct?
Perfect.
Brilliant. So
Yes.
Thank you for having me here. Really appreciate that and the exposure. So I'm Agnieszka Janeczek, co-founder and CEO of Renovos. And Renovos is a UK-based regenerative medicine company that's pioneering synthetic nanoclays for tissue regeneration and more specifically for delivery of biologics. So we all know that low back pain is the main cause of disability in the world. And in fact, every 1 in 10 people are affected by low back pain globally. So it can often lead to invasive spine surgeries in order to regenerate tissue. And the need for tissue regeneration is driven by multiple demographics. As we know, on one hand, we have more active younger people damaging their tissues through sports and trauma. And on the other hand, and predominantly by an aging society. So the global population over the age of 65 is not only expanding rapidly, but it's also staying active for much longer. So in fact, the number of people aged 60 and older is projected to double between 2020 up to 2050. And we know that stem cells within our bodies can regenerate tissues and are triggered by powerful biologics. And BMP2 is a bone-forming protein, quite well known in the musculoskeletal field, and it's most widely used in the field and represents a $1 billion global market, predominantly in spine fusion surgeries. But the current clinical product leads to adverse events due to high doses that need to be applied because the protein is not well retained at the site. So these adverse events include overstimulation and bone growing outside of the skeleton, trapping nerves, inflammatory reactions due to the high doses, again, leading to soft tissue swelling that causes difficulty in swallowing or even breathing when it's done at the cervical spine fusion level. So in many cases, BMP2 still remains the only reliable option for promoting bone tissue formation and fusion and avoiding costly revision surgeries. So we need to deliver biologics such as BMP2 in a precise way. And our solution, Renovite nanoclay gels, offers that precision. So Renovite nanoclay is a de-risk technology. It's based on over a decade of research at University of Southampton in UK, which we spun out from. Renovite's injectable, it's bioactive, it's a degradable matrix that, unlike others, retains biologics. So thanks to that retention, we can dramatically reduce the dose, lower the cost of goods, and improve safety for potent biologic drugs. And nanoclays are actually well-known industrial ingredients. They're used as rheology modifiers. We have refined and validated our composition and formulations of Renovite gels specifically for therapeutic use. So we're translating an industrial ingredient into the medical field because of its sought-for properties. So our nanoclay is synthetic, it's easily scalable, and we have an established clinical manufacturing process and multiple granted patents to date. So our first product, Renovite BMP2, has been granted breakthrough device designation by FDA and is harnessing the power of BMP2 and addressing that $1 billion global market with use in spine fusion. And hopefully the video will work as it explains our first application. So Renovite is injectable, it's easy to use, and it can be easily applied in both open and minimally invasive surgeries, and it remains precisely where injected due to the unique ability to absorb and bind proteins from the blood, which further cross-link the gel and make it much more viscous. So BMP2 is tightly retained within the gel structure, so only cells invading the material get exposed and get affected by the BMP2 in the space. So they form and differentiate into osteoprogenitor and bone-forming cells. And exactly where the material was delivered, that's where the new bone tissue forms. So it acts as a very precise tissue template. Now let me just check because I can't quite get my video to work. On the next slide, apologies. I think I stopped sharing. I'm going to try again, and let's see if that works. So hopefully you'll tell me that it works again in a moment. Is that right? Okay. There we go. I can see it correctly now. Hopefully you guys can as well. So the key properties really of Renovite nanoclay can be summarized in this slide. Obviously, there's many more publications over the years that we had as a spin-out. But the first thing to remember is really it offers a high precision handling. It can easily pass not only through a cannula, as you can see in the picture, but actually a 23 gauge needle, making it suitable for minimally invasive procedures and also even endoscopic going forward. Secondly, it's very well retained at the delivery site. So it retains also BMP2 within its structure until the material biodegrades and is synergistically with new bone formation in its space. And I'll show a handling video on the next slide, and hopefully it will work fine. And finally, because of the lack of diffusion from the material, in fact, lack of any sort of slow or staggered release, the quality of regrown bone tissue is superior to technologies that, in fact, rely on release. So cells actively invade the Renovite scaffold, form high-quality trabecular bone structure throughout the implant space. And the lack of release also leads to other improvements over safety, reduced inflammation in the area, circumventing some of the most common adverse events I mentioned earlier on with BMP2 use in the clinic. So this data is neatly summarized by our Japanese orthopedic surgeon collaborators publication that came out earlier this year. So it serves as a great third-party validation of our technology. And here's an overview of the handling, and hopefully that will work fine as well. So the gel can be combined and is combined and reconstituted with BMP2 solution for simple syringe mixing, as you can see at point of use, and it provides an injectable bone graft substitute material which can be applied both during open and minimally invasive procedures, as I mentioned. So it can be placed inside of that cage that you could see there. I'm going to rewind that again. So it can be placed inside of the cage before the cage is inserted or following the cage placement, depending on surgeon's preference. As you can see, it also allowed for use. It can also allow for use of expandable cages, and you can see it's really well retained inside the cage. So when it's administered into the body further, the material becomes even more viscous and remains at the delivery site. So you can see it withstands a stream of running hot tap water. I can just rewind it here again. So it's really well retained within the cage, unlike a normal gel would behave. And also it's really well retained after exposure to bodily fluids. So you can see that even withstands hot tap water. So our first product offers many advantages over competition, fits within an existing care pathway. We've conducted various external market access interviews with clinicians, with payers. There's a need, an appetite for a new, improved and safer product. In some countries also a willingness to pay a price premium. Reduction in the dosage leads to lower cost of goods, so it will enable us entry into more price-sensitive orthopedic segments, also beyond spine and other geographies, so further expanding the BMP2 market. The injectability of the material allows for pursuing orthopedic indications where BMP2s weren't used to in before. And we know that aging, diabetes, obesity, smoking, osteoporotic patients all impact spine surgery outcomes and increase complications. So novel materials that lead to reliable fusion in the space in all patient populations are urgently required and of interest to clinicians and healthcare systems. There's also a significant growth opportunity in the spine fusion market. Additionally to the statistics I showed earlier, patients are entering orthopedic care funnel at younger age than they had in the past. In fact, 21% of lumbar fusion surgeries are performed on patients below the age of 45, and a further third on 45 to 64-year-olds. So also improved patient confidence and successful outcomes due to new technologies is projected to further increase the demand for spine procedures and increase the market's growth and also M&A potential in the space. However, the RENOVITE is a platform technology compatible with a wide range of healing agents. And even though our first targets are addressing the orthobiologics market, there's plenty of opportunities beyond spine and orthopedics. And this is our pipeline with our mission in mind to help clinicians deliver safe and more efficacious regenerative treatments for tissue repair and enable that wider regenerative ecosystem with the novel biomaterials. So beyond bone growth, cartilage, and antimicrobial delivery, we're also developing proof of concept data on wound healing, cell delivery, and other immunomodulatory applications. That's the team behind Renovos. We have world-class academics, hands-on clinicians that help us develop our material, and also some industry veterans that are advising us on our journey. So we've been endorsed by several organizations and innovation organizations, including also Orthopedic Research UK, a charity and a key stakeholder, and by Biocomposites, a corporate in the medical device space, by making their first ever equity investments in Renovos. So we've been supported by a variety of innovation funding and programs, and we were awarded a Gold Best Spine Technology Award by top practicing spine surgeons via Orthopedic this week last year. So that's my last slide. To date, there is the technology, validated it for pioneering medical use. After being granted the FDA breakthrough designation, we've built further momentum by establishing overseas collaborations, providing yet more external validation to the technology, and translating the manufacturing process into the clean room environment. And we're now raising a $5 million round to progress towards human trials in 2026 for our lead program, but also pursue strategic partnerships and licenses and accelerate the pipeline in other areas. So that would lead to multiple exit options for investors. And just to summarize, really, the orthobiologics assets are viable M&A targets. From 2017 to 2020, they accounted for 9% of all orthopedic acquisitions. And even between 2021 to 2024, the segment jumped even to 20% of all transactions, making it the second busiest orthopedic segment after spine. So some of the larger companies, they're interested in orthobiologics. They're underrepresented currently, so they are on the lookout for new technologies. And that's why it's an attractive space with good exit potential. So it's a great time to invest and support Renovos and our technology. So thank you very much for your time, and I will welcome any questions. Thank you.
Wonderful. Thanks so much. Panelists, take it away. Chris, you're on mute.
Chris, you're muted. This always happens, right, on those calls. So
I think Drew muted me. But anyway, I was going to just mention real quick. So the application in spine, and of course, I'm thinking back to my Infuse days, the way they would use it, not only within the cage itself, but they would also do some fusion in the posterior gutters of the spine. This is intended to be strictly in the disc space, correct?
Yes, so our first indication is interbody fusion. So it will go in the disc space, it will go in the cage, and it will go anteriorly to the cage because that's where you normally place the biomaterial for good fusion. So that's our first indications, but we're looking also at posterolateral fusion, and you can see, our smaller animal data was all from posterolateral models and looks pretty good already. But we have some improvements to do potentially with the formulation when it comes to using larger animal models and humans, but this is also for us on the radar.
Okay, great.
Otherwise, we know that the infuse always had to be mixed with something as well to be applied more safely. And these sorts of sushi and burritos and other sorts of combinations of synthetic and non-synthetic bone to make it handle well. We want to develop one product that handles well in a variety of procedures.
Excellent.
I was curious. Thank you for the presentation. Is your approval just CE mark, or are you also working on regulatory pathway in the U.S.?
So we're mostly focusing on the regulatory pathway in the US just because of where the market is for BMP2s currently. In Europe, because of the cost of the current technologies and because of the safety aspects, the market's much, much lower, and 10 times lower, in fact, than in the US. So our first target is the FDA, and that's where we proceed with the FDA breakthrough designation. This is a novel material, something to consider. Our pathway is obviously a bit more lengthy than a 510K. It's a novel material applied in that space, so we will be going through a PMA route.
Understood. And with talking about the indications from the clinical side, is it just primarily single level at this part, or can it be used in multiple levels, and could it be used in adjacent disc disease after fusion?
It would be great to have the funding to tackle a variety of those indications. Initially, for the kind of cleanest result, we're aiming for an interbody fusion at one level. But we are, what we do want to include those kind of, comorbidities in patient populations that are normally not included in trials. Because it's a BMP2, we know exactly what it does on the tin. It's the most powerful biologic in the space, and we do want to test it, and the patients are actually going to be used in the end as well.
So Agnieszka, you said it's an IDE pathway.
Yes, so we're in discussions with DA currently, and over the past years, obviously, it's a novel technology. They're asking us additional questions about the nanoclay itself and the mode of action. So we've got an ongoing dialogue with the regulators. We are assuming yes. All our data to date shows that it is a medical device mode of action type of material, and yes, we're assuming IDE, and we're assuming, yes, chance also to your earlier question. We are focusing on the FDA approach first, but at the same time, we're making sure that all the data packages that we create and all the regulatory packages that we create are also transferable to Europe and also some other Asian jurisdictions that are of interest to us.
Great. And so you don't have a good feel for what your claim set will be under that regulatory approval, whether you'll have therapeutic claims or device claims or some of each?
Well, I could dive into that, and that could be a very, very lengthy topic, Aaron. But it's a combination product that will be device-led, and obviously the biologic therapeutic would rely on the BMP2 aspect there.
Great.
And I liked your comment too, because you're pointing out a point of where the market's headed. Of course, it's been 25 plus years since Infuse debut, and you hit on the fact of having the 23 gauge needle and the endoscopic spine. And I think that is a really, really valid point as we continue to head that way more quickly.
Yeah, and it's also beyond spine, thinking about procedures. I mentioned procedures where BMP wasn't used before because it is injectable. We're thinking more extremities, smaller fractures, microfractures, where it can be applied in elderly populations where those don't heal that well, right? So there isn't an injectable material that can be as easily placed at the moment that delivers BMP2, right, for those patient populations. So there is a lot of potential with this technology.
Are you going to have any opportunity to gather human pilot data before you lock in your definitive study design?
So we're looking to do a pilot study. That's kind of the requirement at the moment anyway, and what everyone else is doing in that space. So we're raising now to complete our preclinical package, do a larger animal model, and really be ready for that first-in-human study. The first-in-human study we intend to do would be about 40 patients. We'll test 2 doses. That's what kind of everyone else is doing that has a biologic compound. So at least 2 doses of the biologic plus a slightly smaller control cohort. So I don't think you can do any smaller look-see and get any sort of, there's no statistical significance at that level of patients anyway, but at least you're fairly assured about the safety aspects at that point.
Great, thank you.
Is the curing mechanism have something to do with coming out of the syringe and being exposed to air, or what?
No. So there's no timeout. You can prepare it in the theater. You can prepare it at the beginning of the surgical procedure. You could prepare it just before you apply it and apply it straight away after it's mixed. That makes no difference. So it doesn't cure. You don't have a tight working window. You can leave the material. It's going to be fine. The way it works is the more bleeding you have in the area and the more physiological fluids are in touch with the material, that's really where the cross-linking works, and that's when it's in a much more viscous material. But we've designed it in such a way that even, that placement into the cage, you could see the video where you were trying to shake it out from the cage and it still stay there. We designed it in such a way that with appropriate buffers, we can ensure that this is already a much more viscous material before it hits the body to make sure that it stays there.
Very impressive.
Interesting.
Yes, we had a lot of surgeons' feedback on that, and they handled it. So, we're trying to make it as bulletproof as possible for them as well.
So I do have one question from the chat here from Sunil. And he asked, does the delivery system prevent bone growth from BMP2 outside of the intended zone? Will it be safe for the cervical spine?
So we don't have a clinical data yet, so I can't extrapolate it much. But from the animal studies we've conducted to date, we see the bone exactly forms where the material was delivered. So you can see the template over time being replaced by bone tissue. It doesn't leak out, so there's no diffusion in vivo in the body from the material. So indeed, that's the idea, that it's a safe way. And we did studies when we've loaded and overloaded the dose of the biologic, and it was so well retained that we didn't see any of that usual inflammatory reaction around the area which is associated with the biologic. So the retention is truly there, and that's the key differentiating factor there.
Great.
I can ask that one more clinical question if you don't mind. If the surgeon utilizes the patient's bone, how does it stay in place there as well?
So you mean like when they actually use a bone chip or get a bone graft and try and mix it with the material?
Yeah, they take some pelvis and put their own graft in.
Yeah, so this can be easily mixed with everything else. Obviously, for our clinical trial design, we want to differentiate and just test our technology. So we'll be designing it in such a way where they won't need to use any additional bone. Obviously, in larger defects, they would always and why not, right? It's a free material, so they usually do use local bone or bone from elsewhere. It can be really easily mixed with that. So we've done various studies where you can mix other synthetics into it. You can mix patient's bone. It handles really well, and it's in fact, it improves delivery of those materials as well and their cohesion. So it's not a problem of them still needing to use the patient's bone or wanting to, or using something else. For the clinical studies, we obviously just test our material, but we do understand that every clinician has their preference, and they might still wish to mix it with other materials. And it's feasible.
Is there any storage requirement, temperature-wise?
We're designing it to be room temperature stored, obviously, with all the feedback we had from clinicians. We've kind of restricted to cold storage. So that's what we're designing it in mind with. The nanoclay material is really, really stable, low cost, no problems there. It's more on the BMP side and the biologic to ensure that it's stable and that temperature, but it's been done before, the Medtronic kits, right? For example.
Yep.
Mm-hmm.
So will it be pre-mixed in the package, or will the BMP be mixed into the clay in the operating room?
It will be combined in the operating room just because of that cold storage. So it will be similar to what the Medtronic infused kit is and a few others now in development. You will resuspend the protein in a buffer that's given, and through simple 5 times in between syringe mixing, you will load the protein onto the nanoclay syringe. So the nanoclay will be provided as a gel, and that will be ready then to inject.
Great.
Commercial strategy, I'm assuming, then would be, try to create a licensing or acquisition deal before full commercialization.
Ideally, yes. So we know we need clinical data, right? But we're obviously aiming to design the study in such a way that will give us really good, high quality clinical data on the safety and performance of the technology. And we do have aspirations. There were a lot of orthobiologic acquisitions, as you guys know, recently. But also, when it comes to premium biologics, there's not so many out there. And we know that BMP2, it's still used in 30% at least of lumbospinal procedures. When you talk to some clinicians, they say they use it in 99% of the patients. So they know it works. They wanted to use it safer, and they wanted to use across various other procedures. So there will be a need for a premium biologic product, and we hope there are going to be some companies that would like to compete in that space.
Great. Well, thank you again, Agnieszka. That was a great presentation. And there's some additional questions in the chat that you can go ahead and respond to.
Brilliant.
With that, we'll pass it on to our next presenter, Jonathan.
Thanks so much.
At OrthoPreserve.
All right, thank you. Let's see. Okay, everyone see this?
Yes.
All right. Well, hi, I'm Jonathan Schwartz, the CEO and co-founder of OrthoPreserve, and I'm excited to tell you about our revolutionary meniscus implant. So there's about 3 million meniscus injuries in the US and Europe every year, a very common injury among aging individuals and active adults. Common symptoms include knee pain, instability, stiffness and swelling, and ultimately immobility that affects a patient's life. So the current surgical standard of care for meniscus injuries is just cutting out the damaged tissue or a meniscectomy. But when you do this, it disrupts the joint mechanics by removing part of the meniscus cushion, and that can cause recurring pain and high pressure points due to the bone-on-bone grinding, which accelerates arthritis development. So about 1.4 million people every year in the US and Europe get a meniscus procedure, and about 25% of those patients have this chronic pain with no real effective treatments and have to undergo years of physical therapy, injections, and pain medication until their knee is degenerate enough or they're old enough for a knee replacement. So there's really no good options for meniscus replacements that are available, except for allograft, which has its own issues. So our solution to this problem is our minimally invasive artificial meniscus implant that achieves 3 key benefits. The first is relieving those symptoms by restoring the cushion and stability in the joint, allowing for a quick recovery and return to activity after a minimally invasive surgery, and preserving the joint from future damage, including arthritis and total knee replacements. So our goal when creating this implant was to make a device as similar to the natural meniscus as possible. So it's made from a hydrogel material that has very similar mechanical properties to the natural tissue and is self-lubricating for smooth articulation. It's also reinforced with a patented network of fibers that give it extra durability in all directions and allow it to attach in the joint just like the natural meniscus would at the horns and at the periphery to the joint capsule. This fiber design also allows for the implant to restore even contact mechanics in the joint, which is key for cartilage protection. So our implant has several features that other implants in development have failed to implement. None of these implants are FDA approved, and they've had several issues in their clinical trials and early studies. Our implant is the only one with this permanent, fully integrated network of fibers that allow for attachment and prevent common failure modes seen in other implants, such as dislocation seen in new surface and wear between different components causing additional failure in Artemis. It's also a permanent implant, doesn't resorb like menisco fix and turn into weaker tissue that can't survive within the knee joint. So early development on the implant at Georgia Tech, we were able to show that over 15 mechanical properties either match or exceed that of the natural meniscus tissue based on literature values and design inputs. Using that data, we got a NIH small business grant to continue development. Using that grant, we were able to do some knee simulator fatigue studies showing the implant maintained integrity and continue to restore those normal joint mechanics even after millions of cycles at a high impact load. We also developed a minimally invasive surgical technique that just requires 4 small incisions and a 45-minute surgery using techniques most sports medicine surgeons already know how to do. We also did our first animal study in sheep, which demonstrated the implant's ability to restore the sheep's mobility very quickly, protect the cartilage, and maintain its integrity in the animals. Using that data, we got our FDA breakthrough device designation, which will accelerate our development timeline. So from a business perspective, we have the benefit of a large addressable market and a high margin product. Even if we only target those patients that are having recurring pain after meniscus surgery, we're looking at a $2.1 billion market, 70% of which is within the US. After that, we're going to expand our indications as a primary and arthritis preventative treatment as we continue to run our clinical trials after approval to expand to the total market of $8.4 billion. So our implant has all of the benefits that are provided to each stakeholder, which is critical for success in healthcare. So the patients will obviously be a big beneficiary with an improved quality of life by using our functional long-term implant and having a quick recovery and return to work or activity. The providers will like it because it gives them a new tool in the toolbox. There's really no surgical options for those patients that are having recurring pain after surgery. So it gives them a new surgery, very quick, outpatient, ambulatory surgery center friendly. And it doesn't take away from their kind of knee replacement patients because they can still get those later on. It keeps that option open. It'll also give improved outcomes to drive more business to their practice. Payers will like it because it will reduce ongoing payments for conservative treatments and repeats of failed surgeries, and it'll offer a lower cost option to total knee replacements, which can be pushed down the line. So I originally invented this implant while at Georgia Tech for graduate school, and then spent a few years in the medtech industry before coming back to the project after partnering with some orthopedic clinicians who recognized the huge problem there was for this in their practice. We were able to recruit a great advisory board that has all aspects covered needed to get to market and begin driving adoption. So we have regulatory, engineering, clinical, and commercialization expertise within our team. So last year we completed our grant research, got our breakthrough device designation, and we're the first orthopedic device enrolled into the FDA's new Total Product Lifecycle Advisory Program. So what that does is it facilitates engagements with patients, provider organizations, and payers experts to streamline the market access and reimbursement processes. Our current seed round will allow us to complete our remaining preclinical activities and ultimately culminate in an IDE application for our first-in-man clinical trial. We are also participating in the Plug and Play MedTech Accelerator, which is sponsored by Zimmer, and we have a personal ambassador, Zimmer, to help mentor us through that program. So after this round, we will begin our clinical development and raise another couple rounds, each one representing a value inflection point and significant valuation increase until our de novo approval around 2029. At that point, we will do an initial commercial launch through some high-value surgeons to demonstrate the potential of the implant, and then look for acquisition opportunities from strategics. We're also hoping to expand our indications at that time and look for ways to adapt the technology to other joint structures like spinal discs or hip implants. So we do expect there to be a strong appetite for acquisition following FDA approval and early revenue, because our device fills a significant gap in major strategics portfolios for patients that aren't ready for their knee replacements and need another option. So we expect at these acquisition prices, the ROI of our current round will be about 15 to 25x. So we've been able to accomplish everything so far with around $500,000. So we've been very capital efficient. Our $2 million seed round will allow us to complete a GLP animal study, refine our manufacturing, do our V&V testing, and get some additional IP protection, ultimately culminating in initiating our U.S. clinical trials. So thank you for your time, and look forward to discussing opportunities to help us return patients to the activity as they love with our meniscus implant.
Awesome. Thank you, Jonathan. Nice presentation. We'll open it up to Q&A from here.
Jonathan, great presentation. I'll start if you don't mind. I had a kind of a clinical question in a sense. It looks like, at least on some of the depictions that you provided, that you were replacing the entire meniscus on the medial side.
Yep.
And does it require a certain percentage of the meniscus to be, need to be removed, or a large bucket tear before you place it? Or what is the narrowing, I guess, of the indication in this instance?
Yeah, so for our breakthrough device designation, our indication is for patients that have already had a meniscectomy, so part of their meniscus removed, and are still having pain and impairment, or people with like a significant, clinically significant meniscus tear. So like a bucket handle tear where they would have to take out almost all of it, our device could go right in. Otherwise, it would be patients that have had some trimmed out, but are still experiencing some pain and don't really have any other options. Then you could also put ours in.
Understood. And is it good for both medial and lateral side?
So we're just starting with medial, since that's about 80% of injuries. And then once it's approved, we'll 510K for the lateral side.
Understood. Expansion indication.
Yep.
How involved is the surgical technique, Jonathan?
Yeah, so we've done several cadaver labs with my co-founder, Dr. Cyrus Kump, and one of our advisors on the sports medicine side. And both of them have been able to complete it. No real issues. I mean, it was a little bit of learning in the first few, but it uses, common techniques like root repair techniques, bone tunnels, suturing to the joint capsule. So pretty straightforward surgery. We don't expect there to be a huge learning curve, and we were able to complete all of them, within an hour.
Jonathan, are there any relevant animal models that can be used for preclinical testing for this implant?
Yeah, so we've already completed one sheep study, and then our next GLP study is also going to be in sheep. The first sheep study was a kind of more of a pilot study, making sure the implant held up, didn't cause any serious reactions, and was able to get the sheep back to normal mobility within 2 weeks. The sheep model is kind of the gold standard for knees. They have very similar knee, forces and anatomy. So we're going to continue with that model.
Great.
Yep.
And then similar question that I asked Agnieszka, just the pathway along the way to getting regulatory approval. Is there a stage where you'll do preclinical or pilot human data before you lock in your clinical study design for the definitive approval?
Yeah, so through the TAP and Breakthrough program, we're constantly communicating with the FDA, and we are going to do a small 5-10 patient early feasibility study, either within the U.S. or maybe outside the U.S., and then use learnings from that to design our pivotal study.
That's great.
When you look at other technologies as it relates to active implants with new surface and things like that, they've been around forever. And my question along that line is, what do you believe is going to set you apart to where you don't get into the trap that they've been in terms of getting the approval and getting to the commercialization? Tell me your thoughts about that.
Yeah, well, I knew about new surface when I was in graduate school back in 2017, 2016, and knew they were having those dislocation issues, the secondary tearing issues, and kind of designed our implant around the failures that they were seeing. And luckily, they kind of paved the way and taught all the other people trying to make one what not to do. So we made sure we had, our fixation to both the meniscal roots and the periphery. So having this peripheral fixation gives us that extra stability in the joint. We also did a anatomical shape instead of the disc shape the new surface had. So it shares the load in the joint like the natural meniscus does instead of taking that whole load on. Our material combination is also different. It's very sturdy hydrogel. So it has similar properties to the natural tissue. It's not just kind of a harder plastic. And it's flexible. It can kind of mold to the shape of the patient's joint. So getting the sizing completely perfect is not as critical when you have that flexibility. So yeah, we've definitely learned from other people who've tried it, and we think we got it right this time.
Good. I like the confidence.
I assume the implant delivery system is rather agnostic to any scope system that would be utilized by the surgeon.
Yeah, so our cadaver trials, we just use kind of standard off the shelf drills and reamers and everything. So yeah, it's we would like to get our own kind of kit together at some point, but for these initial human studies, we're probably just going to use off the shelf from other companies.
Yeah.
Jonathan, I wasn't clear on your slide. The FDA pathway, is it a PMA pathway or is it a de novo 510K pathway? And how would those differ?
Yeah, it's de novo. So just shorter, slightly shorter than the PMA, obviously longer than a 510K. But yeah, we're going to have to do our clinical trial, but the requirements aren't as bad as the PMA. So it is a novel new device. There isn't anything like it out there. So we can't do 510K, but so we're in the middle.
Right. So are you going to have a control group, and how much patient follow-up is going to be required in the study, or are those things still being determined?
Yeah, so the study is going to be around 150 patients. We will have a control group. We're expecting that to be just meniscectomy, and it'll be a 2-year follow-up.
Great.
Yeah. I was kind of thinking the same thing of when Aaron may have been going with this too, just on your seed rounds. This is a potential recommendation. I don't know what the other panelists would think, but I think it may be more than a $5 and $10 million at Series A and Series B to complete that many patients, and especially with the follow-up.
Yeah. I mean, we did get we've gotten some preliminary quotes from like Micra, and that's kind of how we gauged our round sizes. But like everyone says, you end up spending more than you think. So.
Looks like you've done pretty well with grant writing. I mean, that's something, if that's a skill set in your team, take all the non-dilutive funding you can get with grants.
Yeah, we plan to apply to several more. So when we started the company in 2021, it was just a small team side project for a while. And then I had to learn how to write grants myself. So I didn't get the first one, but we got the second one, and that's what allowed me to move full time, and been doing it ever since. So we're definitely planning to get as many grants as we can, even though that situation's not ideal right now.
Mm-hmm.
Yep.
Just out of curiosity for the group listening and stuff too, what are the mechanics of your note for your seed round?
So
you're doing a convertible notes or?
Yeah, we're doing a convertible note, 8 million valuation cap, 20% discount, and 5% interest.
Okay.
And we have it partially filled, and then some soft circled commitments that are really just waiting on a lead to do the due diligence.
So are you seeing from an investment standpoint, because the market is still squirrely, to use that Southern term, do you feel like the angels are the ones to go to, the family offices? What are you seeing in terms of the hit rate? And for whom?
Yeah, I mean, we're really targeting angels, angel groups, family offices, VC. A lot of the VCs I've talked to kind of want to wait. I mean, there's definitely interest, but they want to wait till we get to the first in human. But definitely planning to follow up with them. But yeah, for this round, our investors so far have been individual angels, and then we're talking to a few groups, angel groups as well, and some family offices, especially around the Atlanta area.
Okay.
A couple questions that came in through the chat. One is, how many anchor points are used to secure the implant? And then follow up to that, any reason that you're just going to start with the medial? That answer was already provided, 80% of the market. Are you planning to eventually, go outside medial? What's the thought process and then the anchor points?
Yeah. So the just like the natural meniscus, anchored at the two horns, and then there's loops all around the periphery. There's about 10 loops around the periphery. So a surgeon can suture, one or two of the loops. We just did one in the animal study. It worked pretty well, but if they want some extra, stability, they can do two. But yeah, like I said earlier, we're definitely planning on doing a lateral after. We really want to focus on the medial at first, and try to get a 510K for the lateral, because that's a much easier path.
Yeah. And then are you going to do multiple sizes, or is there just going to be small, medium, large?
Yeah, so likely about 5 sizes. We are refining those now with our cadaver studies and looking at the literature, scans of patients' knees. But since it is flexible, there's a chance we can either stick with 5 or even less. Don't expect to have or need much more than 5. And eventually we would love to look at kind of high performance custom options, for high performance athletes that have meniscus issues. So those would be kind of 3D printed molds, higher price point, but athletes are probably willing to pay a little more.
Yeah, I was going to ask too on that front is, as it relates to wear rates and testing, I think that is probably something in a lot of folks' minds, especially related to particulate debris. Everybody's being more active. It's just like what you've said, and it's only going to continue. And and I look at that and I just wonder, did you test out to failure? First thing. Second thing is, have you studied the particulate debris that can be generated over a certain number of cycles?
Yeah, so we did not test a failure yet. That's part of this next series of testing. So the FDA just wanted us to test to 2 years of use or 2 million cycles. So that's what we did. Didn't see any failures. We have not evaluated the wear debris in those solutions yet. We have them. We can do that test, but we were going to do a more dynamic test of 5 million cycles and then evaluate the wear debris in those. We didn't see any wear or significant mass loss in our previous test. But the good thing about our material, this hydrogel, is that even if it does wear, the wear particles of this hydrogel do not elicit a inflammatory response that is as bad as polyethylene, like in total knees. So it's a much lower risk, wear particles for our materials.
Okay, thanks.
Have there been other medical devices used in synovial spaces with a similar biomaterial? Just thinking about safety track record.
Yeah. So it's been used as like a cartilage defect repair. Cartiva, maybe you've heard of it. It's had its own issues, but not material rejection related, more kind of the way it was attached and the forces that were exerted on it. So this fiber reinforcement has been used in sutures and angioplasty balloon reinforcement. So both materials have been used in implantable devices.
Great.
Wonderful. Last question from the chat. Any thoughts on the post-op rehab protocol?
Yeah, so likely going to be, crutches for a week, maybe 2 weeks, and then, general PT for the next 4 weeks. So about a 6-week rehab protocol, which is much better than, meniscus repair, which can be months, or allograft, which can be almost a year. And the sheep that we put it in, we're weight bearing on it the next day, complete normal mobility within 2 weeks. We're expecting to be a quick recovery. There's no kind of integration into bone, artificial materials. You can walk on it pretty much right away, but we want to be careful and not tell people to just run around right after.
Yeah. Great. Well, if that's all the questions we have, I want to thank everyone for joining us today. Thank you to Agnieszka and Jonathan. Great presentations. Thanks, Chris, Aaron, Chance, for joining us on the panel. Please reach out to Jonathan or Agnieszka if you have any additional questions, or myself or Drew at MSK Innovations. We look forward to the next event. We have it lined up July 9th. You can RSVP now on our site. Hope everyone has a wonderful night. Thanks again for joining us.
Thanks, everyone.
Thanks, everyone.
Good job, guys.
Thank you.